Hello, my name is Juan José Ospina. I'm a third semester medicine student at Universidad Pontificia Bolivariana in Medellín, Colombia, and today's discussion is about the potent anti-angiogenesis and anti-tumour activity of pegaptanib-loaded tetrahedral DNA nanostructure. This is an article that was made by these Chinese researchers. So in the discussion, we're going to talk about some of the concepts that need to be defined in order to understand the article better. The first one is angiogenesis. Angiogenesis is a word made out of two concepts. Angio, which means blood vessels, and genesis, which means creation. Therefore, it's the creation of new blood vessels out of pre-existing blood vessel cells. Another word is anti-angiogenesis. What this means is that it is the inability to create new blood vessels because the cells lack conditions such as nutrients, growth factors, or the cells themselves are inhibited. The third one is anti-tumour, which is the decrease of the growth of malignant cells. And last but not least is pegaptanib, which is a medicine that is anti-angiogenic. Therefore, it inhibits or decreases the growth of new blood vessels. The general objective of this article is to simply measure the anti-angiogenesis and anti-tumour activity of pegaptanib-loaded tetrahedral DNA nanostructures. There were four main methods carried out in this experiment. The first one was the nanoproduct synthesis. This nanoproduct was made out of DNA by itself. It was composed out of 4 single-stranded DNA molecules attached to the pegaptanib. As you can see here, this is the diagram of the DNA nanostructure attached to this green strand, which represents the pegaptanib. Something important to mention is that this structure had a nanoscopic size, which is the millionth of a millimeter, and the purpose was simply to carry pegaptanib to cells. The second method used was polyacrylamide gel electrophoresis, shorted as PAGE. This is a method useful to separate macromolecules, especially the ones that carry genetic information, such as DNA and RNA. This consists of a gel exposed to an electric field in order to separate the macromolecules based on their size and their charge. As you can see, this is an example of PAGE, and the higher the structure in the picture, the bigger the molecule is. The purpose simply was to check the correct synthesis of the pegaptanib DNA nanostructures, and also to compare the size of the pegaptanib-loaded TDNs against pegaptanib by itself. As you can see, in the fifth lane is pegaptanib-loaded TDNs, which has a greater size than pegaptanib by itself, which is in the 7th lane, which is lower in the picture. The third method were the cell cultures. There were 2 main cell cultures. The first one were the HUVECs, and the second one were the Cal-27s. HUVECs, which stands for human umbilical vein endothelial cells, are simply cells that when they grow, they tend to form blood vessels. The other ones are the Cal-27s. They are human epithelial tongue cells, which have a tumoral activity or a tumoral behavior. In other words, they are cancer cells. The conditions in which they were grown, they had to have nutrients, antibiotics, and a temperature that was similar to our bodies, which is around 37 degrees Celsius. And the purpose was simply to test the pegaptanib-loaded TDNs in human-like cells. The fourth method was the pegaptanib evaluation. In order to do this, the researchers made 3 standards in order to evaluate it. The first one was how pegaptanib binded to cells. So what they did was put a fluorescent substance which binded to the cells that had correctly acquired pegaptanib, and then with a flow cytometry, they simply counted the numbers of the cells that were painted with the substance. As you can see in this image, they simply measured how the number of fluorescent cells in different pegaptanib concentrations. The higher the curve, and the more to the left, the more cells had acquired correctly the pegaptanib. The second standard was the blood vessels length of HUVECs. As I mentioned before, HUVECs tend to form tubular structures. And the more they grow, the longer these tubular structures will be. In this image, as you can see, the one on top are HUVECs without pegaptanib. They tend to form longer structures, and the one below simply would be the HUVECs with pegaptanib, which form shorter tubular structures. And last but not least, the cell viability, which is the number of cells that were alive before and after the pegaptanib. In this image, the one on top is simply the cells which are represented with white dots without the pegaptanib, and the one below, the cells with pegaptanib, which are much less. The purpose of this was simply to measure the pegaptanib and pegaptanib-loaded TDNs, anti-angiogenesis and anti-tumour activity, and the efficiency of all of them. Another thing that has to be mentioned is that all of the cultures in these images were exposed to vascular endothelial growth factors in order to increase their growth. The experiment offered 3 main results. The first one was the tetrahedral DNA nanostructure synthesis. In this image, the one to your left, you can see the diagram of the tetrahedral DNA nanostructure attached to the pegaptanib. Each strand represents a single-stranded DNA which has a different color, and the green strand represents the pegaptanib, which is always attached to the fourth single-stranded DNA. The image to your right represents a page. What you have to see here is that they checked the correct synthesis of the pegaptanib-loaded TDN nanostructure as they checked the size compared to the single-stranded DNAs. Of course, as they kept adding more of the single-stranded DNA, the molecule became greater in size. And as you can see, the pegaptanib-loaded TDN was the greatest or the biggest molecule out of them all, as it is the highest in the picture. Another thing to highlight is the difference in size between the pegaptanib-loaded TDN compared to the pegaptanib by itself. As you can see in the fifth lane, the pegaptanib-loaded TDN is higher and greater in size than pegaptanib by itself because it has more material. The second result showed pegaptanib and pegaptanib-loaded TDN's cell proliferation. In order to understand this image, you have to separate this in four. The first two on top represent Hubex and the first two below represent Col27 cells. The first two graphs to your left represent the medicine at a 250 nanomolar concentration, and the other two graphs to your right represent the medicine at a 375 nanomolar concentration. What you can see is that at 250 nanomolars, neither pegaptanib or pegaptanib-loaded TDNs work, which means that the cell viability kept at 100%. Whereas in 375 nanomolars, pegaptanib didn't work by itself as the cell viability was kept in 100% in both cases, whereas pegaptanib did decrease the cell viability to around 70 or 80%. This means that pegaptanib-loaded TDNs did decrease the blood vessels and the tumoral activity. The third result showed pegaptanib and pegaptanib-loaded TDN serum stability. This simply means how much time the medicine lasts in blood. In this image, it is simply showing that pegaptanib, when it is loaded to the TDN, it lasts even after 12 hours, whereas pegaptanib by itself completely vanishes after 2 hours. In other words, when pegaptanib is loaded to the TDNs, it is much more efficient and it increases its existence. In the discussion, we're going to review some of the passages quoted by the researchers were actually covered by the article. In the first one, it simply says that Cal27s are tumoral cells. Even though they used them, they never showed that they were actually tumoral cells or they had this activity. In the second one, that blood vessels increase the growth of tumor cells and metastases. Even though they try to explain this or to show this, they never fused Hubex with Col27s in order to show a relationship between both of them and how one proliferates the other and vice versa. And in the third one, that pegaptanib simply inhibits some of the signaling pathways. Even though they decrease cell proliferation, they never show which specific signaling pathways are concluded. Molecular biology is very important in medicine because it can specify diagnostics, it can revolutionize treatments, it can revolutionize approaches to classical disorders such as cancer, and it can clarify physiological roles of macromolecules such as how DNA works, and last but not least, how a better understanding of disorders such as how blood vessels increase the growth of tumoral cells. And the article itself, it is mind-blowing, to be honest. This shows how all resources such as pegaptanib, which is an old medicine, with molecular biology techniques which are kind of new, such as TDNs, increase the medicine efficiency, such as curing cancer, such as age-related macular degeneration, and we can increase the medicine extent and we can cure more patients now.